So my project was on the White Valley Active Treatment Site. I worked with the Murraysville Area Watershed. They just got a very large grant from the Pennsylvania DEP to build this treatment site. The main concern was the AMD in the area, the acid mine drainage. They wanted to treat that, which mainly came from the Pittsburgh coal seam. And their main concerns here were the water quality, mainly the very acidic pH, the iron levels, which were very high, and the aluminum levels, which were also very high. All of these affected the fishability of the area, which was declared to be unfishable in the 90s. And it also affected the macroinvertebrates in the stream, which there are very little of. Um, the plan for the treatment plant is to put in multiple areas of treatment to try and decrease the AMD within the future. And then here I have some graphs displaying how the iron has been increasing slowly since the recording of the water quality.
Muddy Creek is an AMD impaired stream in Preston County, West Virginia. It's a tributary of the Cheat River and was actually the largest contributor of acid mine drainage to the Cheat River. So this study was conducted to evaluate the effectiveness of a watershed scale AMD treatment approach over a point source treatment approach. Two in-stream dosers, the Glade Run and the Martin Creek dosers, were built along the stream alongside the TNT treatment facility. And chemical and biological data were collected to evaluate the effectiveness of the treatment. All of the sample sites saw an improvement in pH except for the upstream control site, and average pH increased from 4.3 to 7.3. Mean concentrations of aluminum, iron, and manganese decreased significantly for every site except for the control site. And all macroinvertebrate sampling sites showed increase in WVCI score, meaning that the fish population and biodiversity increased. The Muddy Creek mouth site saw fish populations rise from 0 to 130 after treatment. We were also able to see that although upfront costs were higher for the watershed scale treatment approach, over time the treatment proves to be cheaper. So overall, we saw that the watershed scale AMD treatment method is successful compared to the point source treatment approach.
My name is Kara Meadows, and this is in-stream biological restoration post acid mine drainage remation of Upper Deckers Creek. For this project, the West Virginia Water Institute partnered with the Appalachian Stewardship Foundation to kind of look at a section of Upper Deckers Creek and see if it has potential to, if it's already remediated waters have potential to get even more healthier. So looking at the historical and background context, this creek is in between Arthurdale and Reedsville, and this is a very historical land. Arthurdale was a New Deal town. So in 1933, there was a lot of alterations made with the natural flow of the creek, and you can kind of see in the upper section here, that's upstream of our research site, has very sharp angles, and that is kind of indicative of how this land was rechanneled to make for suitable farmland. The particular research site has been designated as a reserve through the university, so this is university property. In Arthurdale, Reedsville, it's the geology kind of tells us that it has some trouble draining. This particular section of Preston County is in a syncline, so it kind of looks like a ball, and so so it's a really marshy area, and and so basically we're taking several parameters, and we're trying to look at trying to analyze the creek's condition, primarily from the water. We use a YSI meter, a digital calibrator, flow meter. We're also looking at benthic macroinvertebrates. We're using erosion pins to measure sediment loss and gain, and we're also taking cross-sectional surveys of the creek to determine how deeply channelized it is. So essentially, these are some pictures from the field work we've done. When going out into the field, you can kind of see like catfish fry, so that's indicator that the water isn't so heavily polluted. Most of the benthic macroinvertebrates we're seeing are moderately pollution tolerant. And those are diving beetles, slugs, caddisflies, and damselflies. But basically during these like site assessments, we would see that the creek basically has no habitat structures. And going on to the data, we can our data kind of like assures that. So our water data isn't bad at all. It is, pH is pretty good, our conductivity is pretty good, but what our problem is our site is getting a lot of erosion because of the loss of habitat, and it is very deeply incised. So basically our goal here is to install structures to catch sediment and to hold sediment that's there already. And those would be like tow wood structures and live staking along the stream bank, so to increase, basically put in more vegetation and kind of create riffles in the water to oxygenate the water and to create a habitat for fish, amphibians, and help the low pollution tolerant macroinvertebrates come back to the stream. Because our concern isn't with the water quality from the acid mine drainage, but it is, we're kind of looking at that and going, okay, how healthy can we make the stream, knowing that the water itself isn't the problem. So yeah, that is.
Hi, my name is Sarah Nelson, and my research is on the impact of the Richard Mine Treatment Plant on Deckers Creek water quality. So just as a little bit of background, Deckers Creek is an acid mine drainage impacted watershed in West Virginia, with Richard Mine being Deckers Creek's largest threat to water quality. In fact, Friends of Deckers Creek estimates that approximately 292,000 pounds per day of acid mine drainage are released from Richard Mine. Richard Mine at Source Treatment Center was launched in mid-April of 2024. And because of this, Friends of Deckers Creek has been monitoring both upstream and downstream of the treatment center since late 2022. Additionally, WRI has been monitoring the mouth of Deckers Creek alongside 17 other tributaries and main stem sites in the Mon River Basin. So, with all of this data, my objective was to evaluate the impact of the Richard Mine Treatment Center on the watershed, the Deckers Creek watershed. So I took all of this data, compiled it, sorted it, and made these graphs. I was specifically targeting AMD signal parameters, so this could be anything from dissolved metals, total dissolved solids, electrical conductivity, pH, and more. So most AMD signal parameters in Deckers Creek are improving according to these two graphs. These are Friends of Deckers Creek data, upstream and downstream of the treatment center. This is apart from total dissolved solids, which we saw a increasing trend, and this is just due to an excess of salt and ions in solution after treatment. This makes sense. This is normal with acid mine drainage treatment. Overall, this supports that AMD treatment throughout the watershed and at Richard Mine is improving water quality. So a previous study that I was a part of actually statistically examined the 3RQ data from 2009 to 2023. We wanted to see if these trends in the Mon River Basin were statistically significant. So actually, through this study, we found that there was no upward trend in aluminum and no significant trend in iron. And you know, you look at these graphs, you say, oh, well, it looks like iron and aluminum are significantly decreasing. So there are a couple reasons why there are these discrepancies. One of these is that 3RQ, as you can see here, only reports dissolved metals, not total metals. You can see the dramatic changes in the total metals here, but these dissolved iron trend lines look about the same. Another thing that could be impacting that is that this was statistically analyzed from 2009 to 2023, while this data is being analyzed from 2022 to 2025. So whether you can believe it or not, based off of the graphs, you can see that water quality has been improving in Deckers Creek. There have been more fish spotted just days after the treatment center opening. Further work here, I would like to do statistical analysis on the Deckers Creek data once we have a larger range of data over multiple years. But yeah, it just proves the importance of long-term monitoring and acid mine drainage, even in smaller watersheds.
Go.
So for our project, we decided to look at the different tributaries that flow into the West Fork River. We went through Elk, 10 Mile, Boots, Bigamon, Polk, Upstream Polk, West Fork, Weston, Stone Coal, and Jones, plus a few others. And what we found, the main thing we were looking for was fecal coliform, which we found a lot of that in the Polk tributary. We also looked for nitrate and phosphate, which the ones that have empty boxes are because they had so little of each that we couldn't really detect it.
So we went into each of the tributaries with standard field instruments being our Secchi disc tube, which was used to find the turbidity of the water, which is to see how far you can get without, before the water is clear, to see how much sediment is in there. We also had the YSI, which gives you our water temperature, our dissolved oxygen, our total dissolved solids, our pH, and our oxidation reduction potential, as well as our specific conductivity. Those are all just baseline analytes and data to collect anytime you do water sampling, but this specifically looked for the fecal coliform and the nutrients. This was asked of us to do by the Guardians of the West Fork. We found that in the Polk tributary, there was an excessive amount of fecal coliform with also a kind of raised number of nitrate, but it's still within a safe level of nitrate. With that said, this is the baseline analysis of the West Fork watershed, and this is setting up for future replications of this study in each season to come for more volunteers.
We are hoping to resume this throughout next semester and most likely the following summer. So this is just from the very first time, and then for the next times that we work on this, we'll build onto it, we'll see what has changed, what has gotten better, what's gotten worse, try to figure out why that is, see if any of management recommendations are able to be put in place, and if they are put in place, if that seemed to be beneficial.
And with the high fecal coliform levels, it should be noted that more samples should be taken in the Polk to see where the fecal coliform is coming from, whether it's broken sewage sanitation systems, or if it's in a more rural area where people are straight piping their sewage into the stream because they can't afford to be on the sewer system. But that's for further research. This one was just a baseline analysis.
That's about everything. Should we turn it off?
So my study focuses on Deep Creek Lake Watershed in Garrett County, Maryland. This watershed is a man-made lake that was made 100 years ago roughly, and over 1 million cubic yards of sediment have been influxed into this lake. This influx of sediment causes turbidity, decreased water quality, and disrupts biological systems within the lake. And there are 12 coves, and we looked at Poland Cove, which can be seen right down here, which is connected to Poland Run. So the objective was to just map this run and establish areas of concern while identifying sources of sediment. So for methods, we conducted 2 dates of field assessments, and this stretch is roughly 3,750 feet. We categorized it between 50 and 150 feet based on which segments were most similar, and this middle point was where the visual assessments were done and what is mapped. So the visual assessments included a habitat assessment, stream character, and erodibility scores from the Maryland DNR and West Virginia DEP guides for sampling. And pebble counts were conducted on 7 out of the 12 segments. The segments that have no values in the pebble counts were just completely covered in silt, and we decided that it wasn't really worthwhile to conduct a pebble count there. So looking at this segment, all here is a golf course that is currently undergoing some construction, and then up this way we see some just residential area. So the main pattern we noticed was just that the stream was most stable in this like middle section, so closest to the cove and farthest from the cove showed great signs of instability, high bank erodibility scores, especially as seen in our site 11, which we used to compare to site 6, which were our our most stable and our least stable segments to, again, stream character, habitat assessment, and bank erodibility. And overall, more or less diversity towards these 2 ends. And yes, so this data can be used for the Garrett County government to further regulate the sediment influx coming from this cove and pinpoint areas of greater instability.
Hi, my name is Jay, and my research is evaluating the microremediation potential of Pleurotus ostreatus fungus, otherwise known as oyster mushrooms. And so this study was done to inform an ongoing field trial in Buchanan, West Virginia, evaluating if inoculated fungal biomass is able to reduce the metal concentration and increase the pH in a small tributary of Buchanan River called Bull Run on a private property. And so from those surmised results, I created this controlled lab experiment. And so, yeah, what we're looking at is how we can evaluate the remediation potential of these fungi under sort of a complex hydrological regime. A lot of pre-existing studies predominantly look at one pollutant, or they're on petri dish scale, so this is more of a holistic test. And so it involved 24 experimental units, half of which were inoculated, and then of said inoculated and technically uninoculated experiments, we divided them by variables of flow, which was simulated through water change. Half of the treatments were changed once a week, half of the treatments were changed 3 times a week. And we also separated them by the amount of the Pleurotus inoculated substrate in each of the trials. And so within each trial, there's 2 liters of simulated acid mine drainage that was created to best emulate acid mine drainage effluents in the area. And then water quality was measured via a probe looking at pH and conductivity. What we found is initially, when just analyzing the water quality, we saw that the fungi were prioritizing metal adsorption over pH neutralization. So we see a decrease in conductivity. However, we don't necessarily see a strong change in pH in the inoculated systems. Then later, we also decided to do XRF analysis, X-ray fluorescence analysis, on the fruiting bodies, and that was better, and for that we were better able to elucidate the amount of each specific metal ion that was being sequestered by the fungi. And so for that, we found out that certain fungi like aluminum are definitely being uptaken by the fruiting bodies, whereas other metals like calcium and potassium have strong temporal dynamics visualized right here. And some metals like iron and manganese were not necessarily absorbed by the fungus. And so through this we can surmise that there are sort of competing effects working within this process. And that through the general decrease in water conductivity, we know that the metals are going somewhere. However, we're still pending X-ray fluorescence results of the substrate to see how those metals are being partitioned among the treatments. Overall, I think that we are better carving out how we can use Pleurotus fungi as sort of a tool in the collective acid mine drain remediation tool belt, and how the specifically living biomass can be implemented in smaller scale streams with maybe a lower concentration of pollutants in areas where traditional remediation methods like dosers or slag beds are not necessarily implementable. And so yeah, in the future, again, we aim to do XRF analysis of the substrate, and once we surmise what the fungi are optimized at absorbing, we can then compensate with what they're not optimized for with things like biochar. We can also further explore flow dynamics. We do see that the treatment out of the experimental different treatments, the one that performed best was the one in which there was a 1-to-2 ratio of treatment to SAMD, and that was changed 3 times per week. So yeah, in the future, I think it'll be really interesting to look at how we can further optimize these fungi for microremediation of acid mine drainage. Thank you.
Hello, my name is Gabrielle Bush. This is my project assessing long-term water quality trends in the Allegheny and Monongalia rivers. This project is a continuation of a project that began in the summer, I believe. So this is a very small scale compared to the eventual final research project. So in order to evaluate water quality trends, for this poster, we are only looking at the parameter TDS, which stands for total dissolved solids. It's basically a measurement of all dissolved organic and inorganic substances in water, which makes it a good general indicator of water quality. High levels can give us an idea of if there are possible harmful contaminants, and high levels can also affect public water supplies in a bunch of different ways, hindering the taste, the color, a bunch of stuff that we don't want. So there are a bunch of monitoring programs that have been collecting data for water quality. We're only looking at 2 programs. One is Three Rivers Quest, which is through the Water Research Institute in West Virginia. And the second is Ohio River Valley Water Sanitation Commission, which I'll be referring to as ORSENCO. Three Rivers Quest has been monitoring the water quality in the Monongalia since 2009, and then they later expanded to Ohio River, Allegheny River, and the Monongalia River. The ORSENCO program has been monitoring since 1975. Both measure a bunch of different parameters, but of course we're only focusing on TDS. So the general objective is to evaluate the water quality trends by compiling both of the data from these programs. So the methods, I evaluated both of these programs' data. We only looked at 4 total sampling locations, so 2 in the Allegheny and 2 in the Monongalia, 1 from each program. So we compiled these measurements and then created a standardized data set to evaluate these trends. Which you can see here, this is the trend for TDS in the Monongalia River from 2009 to 2005. and this is the TDS concentration in the Allegheny River from 2011 to 2005. As you can see, it's a little bit harder to see here, but in both of these trends, the TDS concentration is decreasing. So that's a very good thing. That shows us that management has been overall pretty effective, and it also gives us an idea of what we can do to maybe better improve things. Another thing that we can see here is that in the Monongahela River, the TDS plot points seem to be a lot more scattered along the trend line, whereas in the Allegheny River, the TDS is a lot more uniform. That could be due to a lot of different things, and that's something that we could hopefully assess in the future as we go along with this project. So in conclusion, this tells us that by compiling this data, it makes it a lot more easier to look at trends such as this, and it also gives a more compatible and accurate idea of trends in the long term. So our next steps with this research is to expand into different monitoring programs, and then as well as look at other parameters aside from TDS. We also hope that with this compiled data, we're able to use these trends to aid in future research, as well as compile all of this information into a story map for public awareness. Thank you.